Zika is a vector-borne disease. Similar to other arboviruses such as dengue and chikungunya, Zika is transmitted to humans via mosquito. When a female mosquito takes a blood meal from a person infected with the Zika virus, the virus can survive and multiply within the mosquito. It is then passed on to another person through the mosquito's saliva when she takes her next blood meal. However, unlike other vector-borne diseases such as malaria, lymphatic filariasis and dengue, we know very little about Zika transmission. The Zika virus has been found in several different species of mosquitoes, but it is important to determine which species are the principal vectors in an area of transmission so that we can use suitable tools to control them. Some of the potential vectors bite people outdoors in the day, but some may come indoors and bite people at night. Tools that target these different behaviours are discussed later in the course. First, we need to know which mosquitoes are involved in transmission, and this process is known as vector incrimination. There are 4 main criteria that need to be satisfied to fully incriminate a suspected species as a vector of Zika. This can be done indirectly by molecular analysis of blood-fed mosquitoes that have been collected from the field, and you will be shown different methods of collecting adult mosquitoes later in the course. Second, you need to know that the distribution of the suspected vector coincides with the distribution of Zika. The third step involves detecting the Zika virus using molecular screening techniques in mosquitoes that have been collected from the field site, and you need to check whether the strain of Zika found in the mosquitoes is the same as that found in human cases. The fourth step is to demonstrate that the mosquito is a competent vector. This involves laboratory studies in a special containment facility. Mosquito females are allowed to feed on a blood meal containing the Zika virus using an artificial membrane feeding system. Over time, the mosquitoes are tested to see whether the virus survives and multiplies inside them. After several days, the saliva of the mosquito is tested to see whether it has the Zika virus. In some studies, experimentally infected mosquitoes are then fed on a test animal such as a mouse to see whether they can pass on the virus. So far, no species have been fully incriminated as vectors of Zika, but the following slides present evidence to suggest that certain species may be vectors. The first isolation of Zika virus was from Aedes africanus mosquitoes collected in Zika Forest, Uganda, way back in 1948. The virus was isolated from rhesus monkeys found in the forest at the same time. A study published in 1969 isolated Zika virus from a different species of Aedes, Aedes aegypti, from mosquito collections made in a variety of urban and rural habitats in Malaysia. Since then, the virus has been isolated from 7 other Aedes species collected in Uganda, Nigeria, Gabon and Senegal. However, Aedes mosquitoes may not be the only genus capable of transmitting Zika, because the virus has also been isolated from Ansonia uniformis, Anopheles coustani and Culex perfuscus. Although Zika virus has been isolated from several mosquito species, the published studies examining vector competence have only used 2 species, Aedes aegypti and Aedes albopictus. One reason for this is that both species are easy to rear in the laboratory. In 1956, Aedes aegypti mosquitoes were the first mosquito species to be infected with Zika virus artificially in the laboratory. Females were fed on virus-infected blood, treated with heparin to prevent it clotting, and through a mouse membrane. The virus was detectable 20 days after they received the blood meal, and the mosquitoes passed on the virus to mice and rhesus monkeys in the laboratory. Later that year, Aedes aegypti mosquitoes experimentally infected with the virus was shown to infect a human volunteer. He developed a mild fever 82 hours after being infected, and the Zika virus was isolated from his blood during the febrile period. In Singapore, a study was performed to determine whether local Aedes albopictus populations could transmit Zika virus if it was transported to that country, and the species was shown to be a competent vector. Virus was present in saliva 7 days after the mosquitoes were given an infected blood meal. More recently, a study comparing the competence of both species using populations originating from Guadeloupe and French Guiana concluded that although both species were competent, they had low transmission rates. Zika virus has been isolated from natural mosquito populations of several species, mostly Aedes genus, but several of the other genera can also be infected. Published laboratory studies have only been performed using Aedes aegypti or Aedes albopictus. Both species are competent, but neither has been fully incriminated. It is too early to say whether one mosquito species is a principal vector in a particular transmission setting, and we should not overlook the likelihood of other mosquito species, or indeed other biting Diptera, being involved in transmission. Further work is required to fully incriminate the vector, and this should involve making contemporary mosquito collections, screening them for arboviruses, and studying Zika infections in the human populations, taking into account the lag between exposure to virus and manifestation of symptoms, particularly with regards to microcephaly. We also cannot ignore other possible routes of transmission. Other factors are important in Zika transmission, be it anthropootic, that means it only involves the vectors in humans, or zoonotic, that means it involves a non-human reservoir host in addition to vectors in humans. People can pass the virus on to another person without vector involvement. The most well-known route is perinatal infection, and that's when a pregnant mother passes on the virus to her fetus. In addition, two other routes have been proposed. The first involves sexual transmission. There have been at least 3 case reports of men returning home from Zika endemic areas, Senegal, French Polynesia and Brazil, to non-endemic areas with high infectious Zika viral loads in their semen. It appears that in one case, Zika was sexually transmitted to his wife, who was living in a non-endemic area. The second involves breastfeeding, following a case report showing the presence of Zika virus in the breast milk in a woman who gave birth to a healthy baby while febrile and immediately breastfed. The relative importance of direct human-to-human transmission versus vector transmission is unknown, but needs to be addressed to direct control efforts.